Hi, welcome to our fifth module. In this presentation, we will compare one of the most important parts of the urinary system, the kidneys. We will also have a short discussion on the renal portal system and the nephron being the functional unit of the kidney. At the end of this presentation, each student should be able to compare the morphology of the kidneys of the different domestic animals, discuss the renal portal system of birds, and identify and compare the mammalian and reptilian type of nephrons. The urinary system is composed of the kidneys, the ureters, the urinary bladder, and the urethra. Among the organs, the kidneys show variation among species. It is also good to point that birds lack the urinary bladder compared to mammals with a very distinct urinary bladder. Kidneys can be classified as unilobar or multilobar with either smooth or lobated surface. Unilobar kidneys are described to have a smooth surface and a single renal papilla as found in dogs, cats, small ruminants like goats and sheep, and horse. Multilobar kidneys with smooth surface and multiple papillae is present in pigs, while multilobar kidneys with a lobated surface and multiple papillae is present in large ruminants. Birds, on the other hand, have kidneys divided into cranial, medial, and caudal lobes. As variation is present only in the kidneys, this module will focus only on the gross comparative notes of the kidneys, the renal portal system, and comparison of mammalian and reptilian type of nephron. In general, the right kidney is always higher than the left in all domestic mammals, except in pigs. Because of this, if you recall the anatomy of the liver, a renal impression is present on the caudate process and the right hepatic lobe of those animals. Before we compare the kidneys of each animal, let us first review the basic parts of the organ. Shown here is a gross and a section kidney of a dog. The renal cortex is the outer layer of the kidney parenchyma beneath the capsule, while the renal medulla is the inner layer of the parenchyma. Here is the renal hilus, where various important structures like renal artery, renal vein, and the ureter is passing through. The renal pelvis is the expanded proximal end of the ureter. This is the one collecting the formed urine from the kidneys to the ureter. Here is a schematic illustration of the kidney to show the other distinct parts. The kidneys are basically lobulated, meaning they are composed of lobules. Each renal lobule has a renal papilla. This is the apex of a renal lobule that drips urine into the proximal end of the ureter. The renal calyces are cup-shaped structures receiving urine from the renal papillae. The kidneys of dogs are bean-shaped as shown here. The renal pyramids fused into renal crest and projects into a distinct renal pelvis. In cats, it is smaller than in dogs. However, the distinguishing characteristic of their kidneys is the presence of capsular vein. This is unique in cats. In sheep and in goats, the anatomy of the kidney is the same as in dogs. They also have the renal crest instead of the individual renal papillae. However, this is in contrast with the large ruminants. Their kidneys are lobated and lacks the renal pelvis. They have minor calyces embracing the renal papillae that lead into the major calyces and into the ureter. Please note that each lobe has its own cortex and medulla. In pigs, the kidneys are also bean-shaped. Externally, the kidneys are the same with others with smooth kidney due to the fused cortex as shown here. However, the medulla is unfused. It has minor and major calyces, thus it lacks the renal crest present in dog. The right kidney of a horse is heart-shaped while the left kidney is bean-shaped. Internally, it has a structure called terminal recess, which are long tube-like extensions that collect and carry urine from the kidney poles to the small renal pelvis. Same with the dog, it has a renal crest instead of the renal papillae. Interestingly, the wall of the renal pelvis has mucus glands which give the horse urine a turbid appearance. In birds, The kidneys lie on the ventral surface of the synsacrum. It is divided into cranial, middle, and caudal lobes. Both renal pelvis and renal calyces are missing. Uniquely, birds possess a renal portal system in addition to the expected renal arterial blood supply and the renal venous return. The system consists of the cranial renal portal vein and the caudal renal portal vein. The system receives venous blood from the caudal body and distributes it into a second capillary system in the interlobular spaces. The renal portal system has been used as a justification for avoiding the caudal body as a site for injection of medications in birds. One reason for this avoidance is that the drugs may be more rapidly excreted when injected here than is desirable due to their being immediately transported to and removed by the kidney. Another concern is that drugs that are potentially nephrotoxic or those that are damaging to the kidneys may be more so when they are first distributed and diluted to the kidneys via the portal system. Now we proceed to the nephron. The nephron is the functional unit of the kidney. Here is a schematic illustration of the mammalian nephron. The nephrons are responsible for urine production, while the subsequent collecting tubules convey the urine to the renal pelvis. Each nephron is composed of several segments. This can be more appreciated in histology and physiology classes, but let us take a quick view of its parts. The glomerulus is a tuft of capillaries where the blood is filtered to form the glomerular filtrate. This is enclosed by the glomerular capsule or the Bowman's capsule. The next segment is the proximal convoluted tubule. Then, the loop of Henle, which is composed of a descending and the ascending limb. Then it will become the distal convoluted tubule, and finally to the collecting duct where the formed urine will be released via the individual renal papillae or renal crest, the renal pelvis or renal calyces depending on the species. Birds' kidney contain two different types of nephron, and one type is markedly different in structure compared to the nephrons of mammalian kidneys. This unique type of nephron is termed a reptilian type nephron and is similar to those found in the kidneys of reptiles. Here is the reptilian type nephron. The glomerulus of the reptilian type nephrons are found in the lobules of bird kidneys, and a renal tubule connects each glomerulus to a collecting duct also found in the same lobule. However, the tubules of the reptilian type nephrons do not include a segment that corresponds to the loop of Henle, and the tubules of the reptilian type nephrons are contained in the outer zones of the lobules where they are connected to the collecting ducts. In contrast, the mammalian type nephrons have the loop of Henle, as shown here, and extend downward into the medullary cones that support the lobules. Again, birds have both mammalian and reptilian type of nephrons. And that ends our discussion on the comparative anatomy of the urinary system.